So you have heard about MRM data from Hasmik. You have heard about phosphoproteomics data from Philip. You have heard about iTrack and related data from Namrata and Monica. So I'm kind of trying to put everything together from the perspective of data analysis. And so I'll kind of refer to what all of them have mentioned. And like I mentioned, you've been at it for a day and a half now, so which means it's time for a quiz. So the question here is, from a data analysis perspective, which of these are fundamentally different? I am going to wait for an answer, even if it takes any guesses? So the first choice is iTrack and SciLac. So I presume people remember what they are. SciLac is metabolic labeling. iTrack is sort of chemical labeling. And there's label-free and label-based. So there's global and targeted. And then there is phosphoproteomics and affinity-based proteomics. There's all of the above or none of the above. Yeah? Yeah, global and targeted. So someone mentioned it. So from a data analysis perspective, so I mean, this wasn't supposed to be facetious. I mean, I was talking to Carl yesterday, and he said, you know, your presentation has two distinct parts, and I think people are going to get lost in it. So I'm trying to kind of set it up so that that won't happen. And the main idea here is that in a global proteomics experiment, which people have also called a discovery proteomics experiment, the idea is that you don't know what you're looking for. You are interested in two different states, like cancer versus control, or two different cell types, or two different, classes, I would call them, from a statistical perspective. So you're differentiated in discriminating two or more groups of cells or tissues or patients or whatever. So you have some groups, and you want to see which proteins are different in those groups. And to start with, you pretty much don't have an idea of what will be different. So here you're going for a fishing expedition. So the idea is that you're trying to cast your net wide and look for things that are different. And you don't have a priori a good idea of what it is you're looking for. So you do what is called discovery proteomics. You can use iTrack and SciLac to label your two classes of interest and combine them and try to see which ones are different. You can use label-free and run set of cancers and set of controls, and then do a statistical analysis to see what is different. So most of the things mentioned here fall into global. So targeted is what Hasmik mentioned yesterday, where you do an MRM-MS analysis. So you know the exact proteins or peptides that you are interested in, and you set up assays to look exactly at those in your sets of samples. So in a targeted mass spec experiment, the important thing is to properly set up the assay and qualify your assay and understand the characteristics of your assay. How good is it? What is the limits of detection? Is the response linear as the concentration increases in the sample you're trying to, of the protein that you're trying to measure? So you want to characterize the assay first in a targeted experiment. Whereas in a global experiment, you just want to run your sample and try to see, look at the list that comes out and say, which do I think is different? So I'm sort of oversimplifying things a little bit, because you could use a targeted assay that you have properly configured and then measure lots of patient samples and then ask, okay, this protein that I thought was different, is it really different from a statistical perspective? So that is going kind of beyond the configuration into the actual use of the targeted assay. But generally, the major amount of time is spent in configuring the assay. So that's kind of why I am doing a little bit of hand waving here. So this kind of goes into, so one of the things you can do is, there's been a lot of tools that have been created for analyzing genomics data. So there's like mRNA expression analysis has been reasonably well established for like the last 15 years. So why can't you just take those tools and apply it to proteomics? To some degree you can, but you have to be aware of what are the challenges in looking at proteomics data. So the data sources are kind of what I mentioned, the two classes. It was here, but I wanted to emphasize that there is really a big difference in how you analyze it. So the two main differences that I see from a proteomics challenge perspective is that In proteomics, when you don't measure a peptide or a protein, it does not mean it's absent. Whereas in a gene expression or like a RNA-seq experiment, to a large degree, if you don't measure a gene, then you can assume that it's either not present or it is present at very low abundance, so you can say that its measurement was 0. In other words, when you don't see something in genomics or gene expression analysis, you can set it to 0 or some very low value. That is not okay with proteomics because of what Carl and the others have mentioned in terms of how the mass spectrometer samples the input material. So it's quite possible that your protein or peptide is actually present at reasonable levels, but you may not be able to measure it because of the stochastic nature of the mass spectrometer. So you have to keep that in mind. When you have a missing value, you can't replace it with a 0 or a low number. The second issue is, you have heard like Philip and others mention that in order to process a few samples, it takes a few months. And so given that kind of a throughput, especially if you want deep coverage and you want to look into all the nooks and crannies of your proteome for some discovery experiment, you're going to spend a lot of time on each sample, which basically means you can't run thousands of samples in a proteomics experiment. Most experiments I've seen use 1 to tens of samples. There are some where if you have reasonably high throughput assays configured like MRM assays for measuring like 200 proteins, those kind of things you can deal with hundreds of samples. But usually it's very few samples. Whereas in genomics, I mean, even the TCGA study has like hundreds, if not thousands of samples for each cancer type. So that's another difference. So because of that, the statistical machinery you employ has to be a little more carefully crafted because you can't assume the standard large sample asymptotic assumptions that a lot of statisticians make and say everything is fine if we ignore a few factors. So in a global discovery experiment, what you generally do is you run some MS analysis using the groups of interest. So it could be case control for cancer or some other disease. It could be some sort of a time series where you have a baseline and then you measure something at 10 minutes and 50 minutes and 1 hour. So you could have something like that. Or you could be interested in multiple disease states. So you could have a control and then 2 or 3 different transgenic mice or through 2 or 3 different mutations you're looking at. So there could be multiple. So basically the point here is that you have classes of interest and you run them in your mass spectrometer. How exactly you run them depends on whether you're using iTrack, SciLac, label-free. So, but you run them, and at the end of it, after you process your data using software like SpectrumMill or MSQuant or the others that you have heard, you will end up with a set of ratios. So the ratios are basically relative to things you're interested in. So the ratio between control and cancer for a peptide, or the ratio between 60 minutes and 0 minutes for some time series you are running. So you have these ratios taken with respect to some reference, and then you usually log2 transform them, because if a ratio extends between 0 and 1, and that is not a symmetric range, between 0 and infinity, and that's not a symmetric range. Things between 0 and 1 are downregulated. Things between 1 and infinity are upregulated. So the upregulated range is significantly larger than the downregulated range. So it's hard to look at the data and work with the data. But when you log transform it, the downregulated range is from -infinity to 0, and the upregulated range is from 0 to infinity. And so we usually look at log transform data. The other reason for using log transform data is that it also makes the resulting data look more normally distributed so that it follows the bell curve a little better. And then we usually normalize in some way. I won't go into the details now because it's kind of tangential. So once you have your ratios, then your goal is to kind of determine which of my peptides or proteins, which are depending on the domain I'm interested in, are regulated. In other words, which are higher in my cancers compared to my controls, or vice versa, based on 2 or more groups that you might be looking at. So finding things that are different is essentially, from a statistical perspective, a hypothesis testing exercise. So you want to say, in my case versus control study, is the log of the ratio statistically different from 0? Because if the ratio is, if the log ratio is 0, then your cases and controls have pretty much the same value of that protein you're looking at. And so the question is, can I set up a statistical test to see if it is different from 0? If you have multiple groups, it's kind of similar, except that the machinery you would use is different for multiple groups. You use some sort of like a ANOVA or a multiple group comparison, whereas here you would use like a standard t-test or one of those things that many people were probably familiar from high school mathematics and statistics. So this is to kind of recap that these are the kind of workflows that you would use, and I think Philip has gone into excruciating detail on this. So I won't belabor the point, except to note that this is the area where we'll be focusing on. So I mentioned that proteomics is a small sample domain. You deal with 1 to 10 samples and so on and so forth. So when you present that in some audience, especially people who are used to like looking at large numbers of samples, the question they ask is it really meaningful to say that you're going to find differential things when you're running 1 or 2 or 3 samples? So for that, this is sort of like a back of the envelope calculation. There are a lot of assumptions that go into this. But the bottom line is that it kind of gives you a flavor for what are the things you can expect to find when you're dealing with very small samples. So here, this column is what we call the coefficient of variation, or CV, for the assay. So the mathematical definition of CV is standard deviation divided by the mean. So it's kind of like a measurement of a measure of the variance of your measurement. So 0.5 means 50% variance. So on average, your measurement varies by 50% for a given peptide or protein. And 0.25 is 25%. I picked these values because usually the technical variation of a label-free experiment is around 50 to 100%, whereas the technical variation of an MRM experiment is sort of like 10 to 20, 25%, that kind of range. So those are the reasons why I picked those values. And now if you are doing a statistical test and you say my p-value is going to be 0.05, so I fixed my p-value. Now what happens if I have 5 samples? What is the minimum fold change that I can detect if I have 5 samples with this p-value and with this measurement error? This is saying I can detect about a fold change of 3. So if my case and control have a peptide that is on average different by about 3-fold with a variation that is bound by 50%, then I can actually find that difference. How did I arrive at this? So this is based on how the t-test works. So if you look at the t-test as calculating 2 confidence intervals for the cases and the controls, then you don't want the confidence intervals to overlap. So that's kind of the back of the envelope calculation that's being done here. So if you translate that into numbers, you say about 3-fold change is what I can detect with 5 samples. But then what is the power? In other words, if I did have a 3-fold change, how frequently would I actually find it? And the power, unfortunately, is about 35%. So if you actually had a 3-fold change in your cancers and controls, and you had only 5 samples, and this was your measurement error, there's only a 35% chance that you would find it.
What's the number of sample pairs? Are they replicate?
Yeah, so sample pairs, I have it as pairs here because you need a cancer and a control. And so I'm just saying that you have cancer control pairs. So in other words, this would effectively be a 10 sample study. So 5 cancers, 5 controls. They don't have to be exact pairs, but if they're not exact pairs, this analysis will change a little bit. It's easier to do the analysis if you treat them as pairs, and so that's kind of why I'm using that terminology. Did I see another question somewhere? No, okay. So now you can say, okay, 3-fold, that's what's happening. Maybe I'm willing to go 5-fold. So I'm willing to find things that are 5-fold different and stop there. So 5-fold or higher. So if you're willing to go up to 5-fold, then you have a 50% chance of finding it if it exists. So the table you can kind of see as your measurement accuracy increases, you can see that your power for detecting 5-fold change starts getting close to 90–95%, which is a very reasonable power. Even highly touted clinical studies many times have power in the 70–80–90% range. So this is nothing laughable. But as you can see, you need more precise measurement. So that's kind of the point. I think the bottom line of all this slide is to say that small discovery samples can actually be useful, if not for definitively stating what is different, at least for saying what your candidates are that you want to pursue. And once you get there, then you can look at combining genomics and proteomics, or you can do network analysis, or Go enrichment, or gene set equivalent of other enrichment analyses, and then kind of narrow in on the groups or peptides or proteins that you want to focus on. But the bottom line here is that with small numbers, you can find useful information. Any questions so far? Okay. So I said you can analyze small numbers of samples, so let's start with one. So suppose you had a single, when I say one sample, I always mean a pair, because you can't have like one cancer sample and try to find what is different with the cancer and a control without having any reference or a control. So when I say one sample, I mean one pair. So you have a cancer and a control sample, and you have run, I don't know, SILAC, whatever experiment to characterize the ratios of many proteins and peptides. Let's say we are looking at the peptide level. I'll get to the distinction between proteins, and maybe I can just mention it here. So as Carl has mentioned previously, there you usually measure peptides in the mass spectrometer. So if you're interested in proteins, you have to roll up those peptides to look at proteins. And how you roll up is a little bit of a tricky issue, as Carl has mentioned yesterday, with grouping, protein grouping, and all that stuff. And for phosphoproteomics, for example, it doesn't even make sense to roll up to the protein level, because you're looking at the phosphopeptide, and the peptide is what you're interested in. So here, when I say ratio, it's usually ratios of peptides, but if you have calculated ratios for proteins, you can use that too. The analysis is kind of agnostic to whether you're using proteins or peptides. So I'll arbitrarily interchange them, and it really doesn't mean much of a difference, except that you have to do different kind of pre-processing in order to get there. So if you have one sample pair and you have a set of ratios that you have calculated, so the black dotted line is a smooth histogram of the ratios that you have. So you can see the histogram in the light gray. The black dotted line is the actual smooth histogram, if you use some smoothing software to do it. So now you can do this thing called mixture model analysis, where you say, I have some histogram, approximate it with the least number of normal distributions possible. So this mixture modeling basically says, what I have is composed of 2, 3, 4, 5, however many number of normal distributions. Find out what they are. So the point of this is to estimate the component Gaussian or normal distributions that make up your smoothed histogram. So here in this case, applying this algorithm basically gives you 3 components, the red, green, and blue components. So you can see that the green is kind of centered around 0. So you can say that is the distribution of components that do not change because they are centered around 0. There is a small hump captured by the red distribution, which is a small set of downregulated peptides. And then there is a much larger hump captured by the blue distribution, which is a set of upregulated peptides. So now you know what your null distribution is. So now you can say, okay, I'm going to draw a line at where the 95% of this distribution is to one side. Anything above that, my peptides are upregulated. So you identify your null distribution, and then you can calculate statistical significance using the properties of a normal distribution once you have that. And then you can assign p-values to your ratios based on all the other p-values that you have seen in this one sample pair. So with just one sample, you can kind of essentially calculate z-scores and then come up with p-values for your ratios. And then you can narrow in on upregulated things you're interested in or downregulated things you're interested in, and then look at the lists or look at some sort of a network analysis or enrichment analysis to see which ones you believe in and which ones you don't believe in. So this is applying that methodology to real data where you have 3 components, the red, green, and the blue. I won't go into the details because the mechanism is essentially the same. So now if you have more than one sample, you can't really do this kind of an analysis, because now suppose you had two samples. These could be either technical replicates or biological replicates, or two replicates that represent the same criteria you are looking at, same conditions. Usually it's better to have biological replicates because you have more variability in a biological setting, as Namrata pointed out. But if you don't, then you can just at least get technical replicates to get some measure of what your variation is. So if you have two of these, then now you have to analyze one sample and you have to analyze the two separately. And then you'll get two different p-values for each peptide or protein you're looking at. And then you have to now combine p-values. Now if you have three replicates, you have to combine three p-values. And combination of p-values is not really that recommended. I mean, you can do it if you don't have any other option. But when you have an option that kind of looks at everything together and comes up with a single p-value, that's much better. And so that is what is done by T and F tests. So we'll look at the T and F tests first, and then there is a variant of those tests called a moderated tests. So I'll get into those too. And these were the moderated tests were designed specifically to deal with like small numbers of samples. And so they have some nice properties compared to the standard T and F tests, which we learn in high school. So what does the t-test do? It basically takes your data, calculates a number which is called the test statistic, and if that test statistic is greater than some value based on the t distribution, you say the null hypothesis can be rejected, which means there is a real difference between your cancer and control or whatever classes you're interested in. It's essentially like taking the number that you calculate, the test statistic, and comparing it to the distribution. So if your test statistic were -1, then it's kind of in the middle of the distribution. So your p-value would be like 0.4 or something like that. But if the value that you get is to an extreme, like t equals 3, then there is very little of the distribution left after that. So you would have a much lower p-value and hence higher statistical significance. So that's kind of the point of this. So how well you can assess this depends on how many replicates or how many samples you have. The more samples you have, the more easily, the more the distribution kind of falls off before you get to a specific value. So the less samples you have, the more uncertainty you will have, and so the higher your p-value will be. And so usually we use this test to see if the ratio or whatever we are looking at is 0. You could do it for other options by just subtracting the number you're looking for, but generally we are looking for whether the difference we are looking for is 0. So if you are interested in a peptide and you're looking at the log ratio of the peptide, this would be a perfect test. The test would ask, is the log ratio 0? And if you can reject that hypothesis, then there is a difference. If not, there isn't a difference. So for comparing multiple groups, we use this ANOVA method where you try to characterize the overall mean of all the groups. So you put all the groups together and calculate the mean. And then you take your specific groups, so your control, your transgenic A and transgenic B, or your baseline, 10-minute, 50-minute, 2-hour time points. So there are now 3 or 4 groups. So for each group, so the overall mean is put all the groups together, calculate the mean. For the next term here, you separate the groups and you calculate the mean. And then what this test does is it says, okay, how different is my overall mean from each of the group means? Is any of the means significantly different from the group mean? If it is, then there is a statistically significant difference in one of the groups. So the F test or the F test is based on this. So the F test is essentially calculated based on the difference between the values across groups divided by the values within a group. So SS stands for sum square. So you want to square it so that negative values kind of don't end up canceling your positive values. If you're off in one direction by 10, it's equally important as being off in the other direction by 10. And so you want to take both into account. So that's the reason for squaring. But essentially here again, you calculate the test statistic, the F test statistic, and compare it to the F distribution here. And it's the same deal again. So if your test statistic is 2 and you have some specific number of samples that determine the degrees of freedom for this, how much of the distribution is left to the right of the number. So that will decide what your p-value is. So the more extreme your value, the lower your p-value will be, and the more significant the difference is. So the one thing you need to note about the f-test is that you can use it to do comparison of multiple groups, but it will only tell you if at least one of the groups is different from 0. So the null hypothesis that this is trying to test is that the mean of all the groups is 0. And if the test is significant, then you know that at least one of the groups is non-zero. You can't tell which one based on the test. I mean, you can obviously go look at the groups and try to figure out what's happening, but based on just this test will not come back and tell you group 3 is different. It will tell you in groups 1 to 4, something is different. So many times what people do is, after doing this, if they want to know which one exactly is different, you can go and do a list of paired t-tests. So you can do, like, if you have 3, you can do 3 pairs of t-tests and see which one is different. You had a question?
So how many values do you need inside each group?
So you need enough to calculate a standard deviation, so which means you need 2 values in each group. Right, so you need 2 numbers to calculate a standard deviation. So you will need two numbers, but if you have a lot of groups, so then you are calculating a lot of means and standard deviations, so you would need more. So that's where this thing called the degrees of freedom come in. So you must have enough degrees of freedom to calculate all the parameters you have. So for each of the groups, you are calculating a mean and a standard deviation. So that's 2 numbers you are calculating for each group. So if you have only 8 data points and you have 10 groups, then you are calculating 20 numbers with 8 data points. That is not a well-defined problem. So the number of points you need depends on the number of groups you have. But irrespective of that, each group must have at least 2 data points so you can calculate some. So it's the same with the t-test. You need at least 2 points. That's why I introduced this for 2 or more samples, not for 1 sample. Because you must have at least 2 values in order to calculate a standard deviation. Otherwise, you can't calculate one. And all these methods depend on the standard deviation. Any other questions? Yeah.
So would that test be, I mean, I'm assuming the answer is going to be no, but would that be used to assess the relative similarity or differences within, like, members of the group?
So if you have a set of groups, then that is what this is doing. It is trying to assess if the groups are similar.
Right, but like, for instance, let's say there are 3 and so.
Oh, you mean the level of difference? So one is low, medium, high kind of thing?
Yeah.
Yeah, you can't do that with this test. Right.
What
Right.
would be the test to do?
So for that, I think I'll show you some techniques towards the end. You can try to come up with like temporal profiles or some sort of like trend profiles that you can do. Or you can many times you can get a feel by visualizing the data. Or if you think there is a specific trend, you can do linear regression. You can do other methods to evaluate trends and look at things.
How do you degree of freedom categorize by replicates versus biological replicates versus technical replicates?
So for this from the perspective of degrees of freedom, technical and biological replicates are treated the same. So what is the disadvantage of using one versus the other? Obviously, biological replicates represent a lot more diversity and variation. So if you could run 3 replicates for the amount of money you have for the project, it would be better if those were 3 biological replicates as opposed to technical replicates. But many times it may not be possible, and so you would end up running technical replicates. But usually, ideally, you would want biological replicates. Many times what people do is if you have like 5 biological replicates and you're able to run 2 technical replicates for each biological replicates, it would actually be good to take the mean of the 2 and then use it as a 5-sample test, because then your mean is more robust, because you've used more data to calculate the mean. And so any variation that you might experience from a technical perspective is kind of minimized and you focus more on the biological variation. So from the perspective of the test, it's just a number. So degree of freedom is 3. That means you have 3 samples. It doesn't matter whether the samples are technical or biological from the theoretical perspective. But from a pragmatic perspective, you would rather have biological replicates.
So for these, you are assuming that your data is normally distributed. And how good an assumption is that?
So yeah, it is true. To a large degree, many of these things assume that your data is normally distributed. And if you have more than like 10 or 15 samples or replicates, then by the central limit theorem or the law of large numbers, you can say that you can assume that the result is going to be reasonably normally distributed. But for small sets of samples, that's a little hard to say. So that's one of the reasons why we take the log transform. So the log transform data is generally known to be a little more normally distributed. The second reason is that people have been using the t-test in a lot of situations, even in situations where things are not really normally distributed. And there are empirical and theoretical results that show that as the data is farther and farther removed from normality, the deterioration of the t-test in terms of how accurately it gives you a p-value is also incremental. So if you had something that was almost normal, the p-values you get will be almost correct. If it was off by a factor of 10, then your p-values will be proportionally off. So the t-test, there are some statistical tests where if your distribution assumptions are violated, then the test basically falls flat and you'll get completely incorrect results. That is not the case for the t-test. That's one of the reasons why, even though you learned it in high school, people still apply it here because it's what is called a robust test. And so the f-test is also to some degree like that because it's based on numbers that are calculated that are similar to the t-test. So in theory, you really wouldn't want to be applying these if you think they're not normally distributed. But in practice, it's not too bad if you apply it. The other issue here is, so what would you do if you really think it's not normally distributed and you don't want to apply the t-test? Do you have any other alternatives? So then you have to go to what are called non-parametric tests. So parametric tests assume that your data come from a known distribution. You know the form of the distribution, but you don't know the parameters for the distribution. You know it comes from a normal distribution. You don't know the mean and standard deviation, but you know the distribution is normal. Whereas when you go to a non-parametric test, you're not making any assumptions. You don't know anything. And so you can calculate p-values based on non-parametric tests. So instead of a t-test, there is this thing called a rank test. So you can do a rank test. But the problem with those tests is that for small numbers of samples, your p-values are going to be very conservative. In other words, you will get much higher p-values than you would get here. So in a way, these tests derive some power from the assumption they make about the underlying distribution. And because of that, it helps you in small sample situations. But if it is a situation where you really want to be sure that the p-values you calculate are correct, then you would do a non-parametric test, or there are permutation tests which will give you exact p-values for the actual data that you have. So you would need to resort to those kind of tests. Any other questions before I go on? Okay. So I mentioned moderated tests in the beginning. So the problem with tests with small samples is that suppose you are calculating a standard deviation with two numbers, and it just so happens that by random chance one of the numbers was off by a little, and the standard deviation was kind of close to 0. And then when you take the ratio, then the ratio is going to be unnaturally large because your standard deviation was slightly off because of some random error. And that was not offset by the fact that you had a lot of measurements. And so when things like that happen, you call the test statistic as being unstable because small changes in your data can result in large changes in your test statistic. If that happens, then it's not a good thing because in one case, you would have had a statistically significant result, and if you just changed one peptide by a small amount, then the statistical significance definitely goes away. So you don't want situations like that. So in order to address that, the people in the gene expression analysis domain have come up with this moderated estimation procedure. So here, basically what you do is you use a Bayesian method to kind of say, based on all the data I've seen, I think my standard deviation is going to be X. And then I go and look at the specific peptide and say, okay, how close is this to X? And how robustly can I estimate the standard deviation based on the number of samples I have. So let's say the overall standard deviation was 1 for the whole sample set, looking at all proteins and peptides. And then you go and look at a specific protein, and that standard deviation for that protein was 10. So it's off by a factor of 10. So it could be a really regulated peptide. But if you had only 2 measurements, whereas the overall standard deviation was based on 10 measurements, you would say, well, I would believe 10, but let me weight it down a little bit because it's based on too few points. But now in another experiment, if you had the same difference of 1 and 10, but the 10 was estimated using 10 data points, you want to give it a lot more weight. So that's kind of what this does. So S naught is basically the standard deviation of the entire population of proteins or peptides you're looking at. SP is the standard deviation for your specific protein or peptide you're looking at. And you calculate these parameters D naught and DP based on Bayesian estimation from your whole data set. They are essentially weights that are learned from the data that you provide. And so what this does is it kind of moderates your variance. So when your variance is based on very few values and it tends to fluctuate a lot, this kind of tends to rein in the variation in the standard deviation and makes the test statistics that you calculate more robust. So that's kind of what the moderated aspect provides. And so we use this in order to account for the small samples small numbers of samples that we have. And the other issue with using moderated statistics is that the p-values degrade gracefully as you have more and more missing values. So this is a kind of, the p-value is 0.3 when you have only 5 actual values you use. So let's say you had 25 measurements you are looking at, and in one case 20 measurements are missing. So your p-value for the same difference would be 0.3. But as more and more values become available and are not missing, you can see that the p-value drops fast, and when you have 10 values, you are already reaching significance. So this kind of shows that you can use this to deal with missing values. Remember in the beginning I mentioned missing values is one of the key things that we have to keep in mind because you can't just put 0 for them. And so here, if you have missing values, you can legitimately deal with them, leave the data out only for those peptides or proteins that have those values missing, and as the values become available or for other proteins for which values are already available, you can achieve significance and kind of capitalize on the more numbers of samples, more numbers of more data that you have for that peptide or protein. So are there any questions up to this point? Then I'm going to kind of slightly switch gears and look at a different issue. Okay. So the next thing I want to very briefly mention is this thing called multiple hypothesis testing. You've probably all heard of the fact that you have to quote unquote correct your p-value when you do t-tests or whatever in genomics and proteomics and so forth. And so the idea is that when you use the t-test, for example, you calculate a p-value. And if your experiment had like 20,000 phosphoproteins that you're looking at, phosphopeptides that you're looking at, like Philip or Namrata mentioned, then you're doing 20,000 t-tests. And so you need to be aware that if you do that many tests, just by sheer chance, something will come out to be significant. And so you have to account for it. And a good example that I found, I don't know, maybe I found this on Wikipedia or somewhere. So a pretty good example is, so the multiple testing problem is the potential increase in false positives. When you, in other words, you think something is significantly different when it is not, simply because you're just repeating a test over and over again. And so an example for that is, suppose you're flipping a coin, a fair coin. So it's for statisticians, a fair coin is I don't know, standard term they use. That's a coin that when you flip, on average gives a probability 5 of falling heads and probability, sorry, not 5, 0.5 of getting a heads or a tail. So you want to assess whether a coin that you got from someone is fair or not. And for that, you test the fairness of the coin by flipping it 10 times. So you flip the coin 10 times and you observe how many heads and tails you get. So if you use a 0.05 p-value, then this is basically saying if you observe more than 9 heads, your coin is biased. So you flip the coin 10 times. If you see 9 or 10 heads with a p-value of a 0.05, you're going to say my coin is biased. So if you do this once, you might get 5 heads, 6 heads, and you would say it's not biased. If you got 9 or 10 heads, you would say it is biased. So now if you went to a bank and they gave you 100 coins to test. So you repeat this, flipping each coin 10 times on each of the 100 coins. So I won't go through the calculations here, but the bottom line is, because you repeated this many times, there is a 34% chance that you will deem that there is at least one coin that is biased. So even though there were no biased coins, there is a 34% chance that you will come up with the conclusion that at least one coin is biased, simply because you did this 100 times. So that's kind of the reasoning behind multiple testing, and you want to correct for this. I mean, this is just 100. What about 20,000? Or in the genome world, it's 25,000. Ubiquitinome, 5,000. So you're looking at thousands or tens of thousands of tests. And so you really have to correct for this. Otherwise, you're going to end up with a lot of things that seem very rosy, and you will be very disappointed. Well, actually, it's the other way around. 0.34 is that all coins are deemed fair. So that means you have a 66% chance that some coin is deemed unfair. So in order to do this, the most straightforward thing is what people call the Bonferroni correction. So you want a p-value of 0.01, and you're doing 100 tests. So you set your threshold 100 times lower. So instead of a p-value of 0.01, now you use a p-value of 0.01 divided by 100. I can't think how many zeros it has, which is why. So basically, you make your test more stringent, more and more stringent, depending on how many times you're repeating the test. The main disadvantage, this is a perfect thing. I mean, if you can do this and you get the things that are significant, you're golden. This is kind of the most conservative multiple testing correction. And the problem with that is it is the most conservative correction. And in things like proteomics and genomics, where you do a lot and lots of tests, many times if you apply this, you'll end up with all p-values being 1, nothing significant, nothing anywhere. So that is primarily because the Bonferroni correction is meant for tests that are completely independent. The correction is really true only when all the tests are completely independent. In other words, the 2 genes or the 30 genes or the 300 genes you're testing are all independent, which is never the case in genomics or proteomics, because there's a lot of interrelations between the expression of one protein and another, or one gene and another. So this condition never applies, and it results in values that are way too conservative. So in order to, there are many ways that you can compensate and come up with multiple testing correction that is more balanced. And one way that people commonly use is called the Benjamin-Hochberg false discovery rate correction. So here what you do is you kind of sort all your, sort all your p-values, and then you kind of decide what your final p-value, I won't go into the details, but you decide your final p-value based on multiplying the sorted p-values by some factor, depending on how many tests you are doing. So generally this results in reasonable correction, and in most cases people use this. It's relatively simple to implement. It's just a couple of sorting exercises, and there's like code that you can write or code that you can find that will do this for you. So I've spoken about moderated T and F tests and multiple testing correction for those. So we have in GenePattern, I don't know how many of you are familiar with GenePattern. This is a suite of software tools created by the cancer informatics group, and you can actually very easily add new programs and modules to it. And we have taken the moderated F-test and T-test module and added it to it. It's called moderated tests. If you're interested in using it, you can find it at the GenePattern beta.broadinstitute.org site for now. But if you're interested, I'm more than willing to help you navigate this. But the input data will be like this. So you have your, this is like phosphopeptide data. So you have phosphopeptides. You have the log ratios for replicate 1, log ratios for replicate 2, and log ratios for replicate 3. And you can see that some of them are missing. For some, 2 values are missing. So for this one, you won't get a p-value at the end. But if there is more than 2 available, you will get a p-value at the end. And if you have more replicates, you'll get correspondingly stronger p-values. So you take a CSV file like this and stick it into the input file column, and you can pick T or F test, depending on whether you have two groups or more than two groups that you're comparing. And when you hit enter, it'll run through and it will create a table that will give you the nominal p-value, the corrected p-value. You can also specify what corrected p-value significance you want, and based on this will actually draw a plot. So this is for a two-sample case where you have replicate 1 along the x-axis, 2 along the y-axis. So this shows the histogram of the ratios of the individual. These are called marginal histograms. So this is for replicate 2 and this is for replicate 1. And when you combine them, this is the scatter plot. The red dots are significantly regulated, up and down regulated. The black ones are not. So you can see that if there is a change in sign, so here it's positive in one whereas negative in the other replicate, it won't achieve significance because there is too much of variation and too much of difference. But if they are consistent, so both are positive and both are sort of positive to the same degree, then you will get significance. And the red dots are based on this number that you set. If you want to be more stringent, you can set it to 0.01 or 0.001 and you'll get fewer red dots here or vice versa. If you have 3 replicates, it will plot something like this, where you have the marginal histograms along the diagonal. And so each one this is called a pairs plot. So this is the plot of replicate 1 versus replicate 2. This is a plot of replicate 1 versus replicate 3, and so on. And these provide the correlation numbers for the corresponding plots. If you have multiple groups, you will get one such figure for each group. So this is for group 1, this is for group 2, and so on. And the interpretation is essentially the same. So here the red dots are decided based on the F test, whereas here the red dots are decided based on the T test. And we use moderated versions for both F and T. So if you have more than 5 or 10 samples, so let's say you have an MRM assay configured, you have 300 cases and controls of breast cancer samples, you run them all on your MRM assay and you have measured 200 proteins in those samples. And you want to do more detailed analysis. So then now you can, because you have larger sample numbers, you can use pattern recognition methods and other standard gene expression analysis tools and other machine learning tools. So we have a collection of these in GenePattern. These are easily available in R, SAS, many other statistical packages, and you can easily access them and use them. You can plot heat maps, you can do classifiers, you can do clustering, and so on and so forth. And someone asked in the beginning, well, if you have trends in your data, how would you see it? So one way to do that is clustering. So this is an example that shows fuzzy clustering, where each point can belong to more than one cluster with some probability. And so the probability is shown by the intensity of the color. Red means it's higher probability that it belongs to that group. And as you get into more and more off-red colors, the probability of that belonging into this group is less. But if you do this and you have 9 clusters, you can see that if this were a time trend, right? So you're looking at time points. You can see that this one starts off high and then drops, whereas this one starts off low and then goes up. So now you can go back, see which proteins or peptides are doing that, and then see if they fall into pathways, or you can continue with your other analyses. So as I've been mentioning, you can apply this to peptides or proteins, and I think I commented on this before, so I won't go into that again. The one thing I want to emphasize is many times people come back and say, okay, I set my adjusted p-value to 0.05. There's nothing significant. So what do? Do I just throw away the experiment I spent $100,000 on and 6 months in the lab? Then you don't have to. So especially with small samples, the goal of applying these tests and corrections and calculating p-values or fold changes is to prioritize the results. So if at the 0.05 level you don't find anything significant, but you look at the 0.25 level and you find 10 things, and you know that 5 of those are really correct because you know what pathways are activated, that's fine. Simply because you have smaller numbers of samples, And simply because Steve Carr's group is so phenomenal that they can measure hundreds of or thousands or tens of thousands of peptides doesn't mean that you end up with nothing at the end, because you don't have statistical significance. Simply because there were lots of tests that you did or very few replicates that you have, you might end up with things that do not cross statistical significance. But you can still use the p-values for prioritization. So you can prioritize the values, look at that. Let's say you're interested in, or you have the ability to look at 25 or 50 things. So you can just use the p-values, sort by that, and then use it. Or sometimes people sort by a combination of p-values and fold changes, and then use those results. So simply because something doesn't come out statistically significant doesn't mean it's a complete wash. It just means that the signal isn't as strong, and the numbers of samples you have and the number of proteins you're measuring doesn't quite support full statistical significance. But then you can use it as a prioritization mechanism. And the kind of accompanying statement with that is that you now really have to use those peptides or proteins and do further analyses, either sort of network pathway or enrichment analyses or MRM validation or other genomic support or actual lab experiments to kind of go and figure out which ones are you tend to believe in. So this is kind of the point where I switch from discovery-based to targeted analysis. So in discovery-based, like I mentioned in the beginning, you're interested in finding what's different. How do I get there? How do I assign statistical significance? What do I believe? And how do I deal with it? Now, if you're going to do targeted analyses, then you're going to do this class of experiments called MRM-MS analyses. And here, primarily, you want to be looking at, so that an MRM-MS assay measures a single peptide or a protein. And you want to be able to say how good your assay is in terms of linearity, limits of detection, how low can it detect in terms of the samples that you're going to provide, things like that. So you want to kind of qualify your assay to begin with, and then you want to apply the assay to samples. So for that, I think you have seen this flow, or Sue is going to talk about it if you haven't. This basically says how you run an MRM-MS experiment. I won't go into it, it's just a placeholder. So to analyze data that comes from MRM-MS experiments, we have this tool called QuaSAR. And QuaSAR essentially calculates a lot of statistics, visualization, and provides a lot of information for you to look at how your assays are working and what you can say about them. So you would start with data from the mass spectrometer. You would process what is using some tool, either a vendor provided or there's third-party tools, which like one is called Skyline. I think Sue might talk about that let you process MRM-MS data. Basically, what you will be doing here is you will process the raw data files, and you will do peak extraction and what is called peak integration. I think Hasmik mentioned peak area ratios yesterday. So those ratios and stuff will be calculated in this tool. So once you have the ratios for all your samples and concentration points that you're trying to assess the performance of your assay, then you would get into QuaSAR. So QuaSAR is essentially this block around collection of algorithms. So one of the things you want to do is quickly, it has multiple components. One is doing calibration curves. It can also find interferences in your MRM transitions. I think Hasmik covered this briefly yesterday. You can quantify your limits of detection and quantification, and you can quantify the precision of your assay based on the coefficient of variation. So each one, I'll just spend 2 minutes on all of them. So audit is one of the tools that we have here that can figure out if you have an interference. So an interference is where when you have a transition that you're trying to measure, so a transition consists of two masses that are combined, right? So there is the Q1 mass or the precursor mass. And when you fragment the precursor, you get multiple fragments, and you're going to look at one of those fragments. So a transition is a combination of a precursor mass and one of your fragment masses. And you want to measure the area or the peak area under your specific fragment mass. So when you're doing that, it's possible, especially in complex samples like plasma, that something else also has the signal at exactly that same mass. So both the Q1 and Q3 masses match, and their retention times are also reasonably close, that one contributes to the other. And when that happens, you want to be able to tell that that is happening, because otherwise your quantification will be off, because that one transition was affected by something not other than what you were measuring. And so to do that, we calculate. So if you are measuring 3 or 5 or some numbers of transitions, we calculate these things called relative ratios. These are the ratios of the intensities of one transition to another for a given peptide. And when you do those, you can apply a statistical test to see when there is an interference. And if there is an interference, this tool will flag. It's also possible that in some cases the coefficient of variation is too large. Remember, I told you that we expect something around 15–20% to be like the achievable coefficient of variation for MRM analysis. If it goes beyond that for whatever reason, the peptide is not behaving well, or there is some other issue with your chromatography, then you want to be able to flag those too. So audit will flag both kinds of issues. Calibration curves, I think Hasmik covered this yesterday. We plot linear calibration curves, but we visualize in the log domain, so we can see all the things as you can see here. So this is the linear calibration curve, and this is the log plot. So you can see all the points. Here, many of the points are kind of cluttered around at the bottom because you are on the linear scale. And we fit calibration curves using robust weighted regression, because as the measurement values go up, the measurement variance also goes up. So you want to account for that using weighted regression. And the software will also calculate confidence intervals and kind of provide you a table with those. Limits of detection and quantification, this is like a domain unto itself. I won't go into details except to say that this is kind of the limit of detection is the lowest value that you can confidently detect in your sample, lowest amount. And the limit of quantification is the lowest that you can quantify. So the difference is that by the time you are at the limit of quantification, your coefficient of variation has dropped, so that you are measuring it in a very precise way. With just detection, you are happy if you can say that you have seen it confidently. Whereas with quantification, you should have both seen it and measured it well enough to quantify. So these two are very important numbers for any assay, because you want to know if you're going to measure some protein in plasma, for example, and your assay has a limit of detection or quantification that is much higher than what would be found in a sample that you're going to get, then it's a useless assay. So this is a very important number that needs to be calculated. We usually calculate it using a blank sample and a low concentration sample. Again, I won't go into details there. I can talk with anyone if you're interested offline, but we have several methods to calculate it, and the most common method we uses the mean and the blank sample. So again, QuaSAR is available in GenePattern. You can plug in your input table and press a button, and you will get all the results and plots. So some examples of what you will get are calibration curves. So this is a calibration curve where you have 3 transitions. All of them are almost along the diagonal. All of them are very close. And you can see the data points. There is some variation here at the low concentration level, but beyond, so the limit of detection is somewhere around here. Beyond that, it's all very nicely lined up along the diagonal. So the top is from a peptide that is well-behaved peptide, and is a peptide that you would definitely use to quantify that protein. Whereas the bottom, you can see that there is more variation in the slopes of the 3 transitions. The values are all over the place. The limit of detection is probably way over here. It's too high to be of much use. You can see these lines here show that those transitions have interferences. So this is a peptide that you might want to consider twice before using it for quantification. So if you have an assay that you're trying to set up for like 300 peptides, QuaSAR will come up with 300 plots like this. So you can just quickly glance through them, pick out the ones that are problematic, and then focus on them and keep the rest. So it's a tool to kind of accelerate the assay development process. So these are other examples from QuaSAR. So the LOD, LOQ table will give you those values. The regression table will give you the slope, the errors of the slope, the intercept. So it will basically give you the regression line and intercept. The audit table will tell you which transitions or which samples have interferences. And then the CV plot, so this is as concentration increases, you can see that the coefficient of variation actually drops quite well. So this is kind of an ideal situation which you would expect. At very low concentration, it's hard to measure, and so you have a lot of variation. As you go higher and higher, you can measure better and better, and your CV drops. So audit will create plots like these if you have replicates for the samples and concentration curves that you have. So we are done. So this is a closing quiz now to keep you guys on your toes. So an MRM-MS experiment measures the level of a protein in 100 breast cancer and 120 control samples. What do you need to measure it, to analyze it? So one thing I kind of forgot to mention here is that so this mostly what I have talked about in QuaSAR is how do you get measures of how good your assay is when you're developing the assay. But once you have developed the assay and you have run the assay on various patients, you can actually process the data in QuaSAR again, and you will get measurements of CV, coefficient of variation and mean for every sample that you have measured. And so you can use that to kind of see what the various measurements are in your sample for the peptides or proteins you're measuring. And so this is more a facetious quiz, I think. So in this case, you would actually need to set it up using QuaSAR and then analyze the data that comes out of these measurements also using QuaSAR. But now if you're interested in, so let's say you measured 200 proteins and you want to know if any of them are actually statistically significantly different between the cases and the controls, then you would use some sort of a moderated or some other test to actually look at that. So this is where in the beginning I mentioned I'm kind of hand waving a little bit. So even though this is a targeted experiment, when you start applying targeted experiments, you may apply it to find things that are different in your samples of interest or groups of interest. And if nothing works, find a statistician. So I went to a conference recently, a year ago, and there was a person who was giving a talk. So she was from England, and she was from a traditional biology department. And she goes, well, we were trying to analyze this data very hard. We had a lot of trouble. And then there was this guy who lost his way and came to our lab to ask how to get to an auditorium. We found out that he was a statistician, and we locked the doors. And that's how they found their statistician. So I don't think it's that bad here. I think you can probably find way more statisticians than you need here. If not, talk to Steve Carr. Some references. So if there are any questions,